Great.
in%.
All right. Well, I'm very, very honored, thrilled, and happy to have Brendan Pinto with us today. Thank you, Brendan, for accepting our invitation. I'm going to talk very briefly about Brendan and his research. Brendan is originally from Sioux City, Iowa, which I had the pleasure to see. I had the pleasure to see his state and his family. He obtained his bachelor's working on population genetics on North American native wild rice in Morningside University, and he switched groups. And then he worked, and he did his PhD at Marquette University in Milwaukee, Wisconsin, working with Tony Gamble, working primarily on studying sex chromosome and speciation in neotropical leaf litter geckos. And then that's where we became good friends. We were lab neighbors. And I had this, to me, one of the most amazing things about Brendan is that he has this kind of like photographic memory. He never took notes on any classes. He never took notes on any talks. And he could recall the talks years after with amazing capacity. We had a great time in Milwaukee. And during that time as well, he became research associate in the Department of Zoology at the Milwaukee Public Museum, and he still holds that position. So in pursuit of a better understanding of the role of sex chromosomes and sex determination in animals, Brendan now participates in excellent collaborations beyond lizards and snakes, including insects, bivalves, humans, non-primate humans, and among other groups. So Brendan is currently an assistant research professional at the Center of Evolution and Medicine in Arizona State University. And he also works as an early career counselor of the American Genetics Association, and is also the leading organizer of the Open Consortium of Squamate Genomics. And I'm sure he's going to talk about that at the end of his talk. So thank you, Brendan. We're going to leave questions for the end, and I'm going to interrupt you when we have 10 minutes to 12 for questions. So thank you, Brendan, and take it away.
Awesome. Thank you, Sarah, for that introduction. I am stoked to be here, albeit virtually, to tell you guys about some of my work. And as Sarah mentioned, we go back in graduate school, and I would like to say that I am severely jealous of your department because since Sarah has joined you, I know that you've gained an amazing colleague, and I'm jealous. So without any more about that, we're going to talk about lizards the rest of the talk here. And that's my bread and butter, so I'm really excited to tell you about it today. So before I talk about lizards, I have to thank the people who let this work, allow this work to happen. I've never published a single author paper. I don't plan to. These are the people who make the dream come true for me, my collaborators, and I especially want to highlight my PhD advisor at the bottom here, Tony Gamble, and at the top, my postdoc advisor, my current advisor, Melissa Wilson, who've taught me way more than I could possibly tell you about not only lizards, but sex chromosomes, which is the other topic of today's talk. So really briefly, I want to give you an overview of sort of the four horsemen of my work. And I especially want to highlight this paper because it was the first one from my PhD, and it's the only paper with a species that I've studied that occurs natively in Colombia. And so that's a one that's very close to my heart. But I'm not going to talk about it today, unfortunately. Although I tried to make it fit the mold, it didn't quite work. So another sort of facet is interesting genomic and molecular underpinnings of complex phenotypes, such as adaptations to different light environments. And then naturally sex chromosomes, generally in geckos, but I'm going to talk about a few other groups and some future work outside of geckos today. And then sort of more broadly, genomics and lizards, and I'm going to talk about this today, certainly both of the things on the left half of the screen I'm going to be talking about more extensively, but generating community resources to enable folks to be doing this work without having to have access to all of the things that we take for granted at a place like Arizona State. And without any further elaboration here, let's move to the topic of today's talk. So today I'm going to tell you about sex chromosome evolution in squamate reptiles, or the lizards and snakes. To do that, I need to tell you a little bit about a lot of things. And some of those things are topics such as what is sex determination and sex maintenance. And we're going to stay with invertebrates today, so apologies for plants and insect folk. Squamates, why are they a good model for sex chromosome evolution? And why is genomics in squamates important? Why am I even talking about it today at all? And then to sort of get at these topics at a more fundamental level, I'm going to tell you two case studies. One within Sphaerodactylus geckos from Puerto Rico. It's part of my dissertation work that although Sarah's heard about before, he's never heard this version, so that's always fun. And then I'm going to tell you about the crocodile lizard endemic to southern China and Vietnam that is very endangered, and hopefully our work will help save that species. We can cross our fingers. But the thing that's, again, tying these, the rug that's tying this little room together is that both of these projects would be unable to be accomplished without the incorporation of a high quality reference genome. And so today, that's sort of the topic of the talk, is genomics being incorporated into sex chromosome research and what that enables us to do. And so dropping back before we get forward, starting over, from the bottom up, we all know that DNA is the blueprint, quote unquote, for life. These four base pairs are wrapped up into physically linked molecules called chromosomes, and all vertebrates, for instance, possess these. However, we share most of the same genes or protein coding regions, but the organization of those genes in the genome itself differs greatly, and that's group specific. But if we took like a simple example, such as the Z chromosome in birds, and we mapped it across these other species on the phylogeny, and just to see where this linkage group or this evolving linked molecule ends up, we'd see in reptiles, it's a single piece, but as an autosome in all other reptile species here shown, and then it's broken apart in mammals. And that's about the extent I'm going to talk about mammals for the rest of the talk, hopefully. But what's important, because we're talking about reptiles today, is that these linkage groups are very conserved within reptiles specifically, which allows us to answer a lot of fundamental questions, even with relatively small amounts of data. So getting into sex determination, what is sex determination? So organisms that want to basically fully utilize recombination as a mechanism for passing on their genes, right? You need some way to bin some individuals as males or sperm carriers, and females are egg carriers. And this can be done a variety of ways. In lizards and snakes, for instance, this is done two different ways. One is genetic, and this is what I'm going to be talking about today. The presence or absence of a specific locus in the genome initiates the sex-determining cascade to become either a male or a female. And this can also be done environmentally, and the most famous example of this is temperature-dependent sex determination, where the environmental temperature of the egg during a critical point in incubation determines sex. But this is super variable across the tree of life. As you can see, these color-coded pie charts here are outdated sort of estimates for what these tips are doing as far as sex determining goes. And you'll see that on the far left here, mammals are pretty much doing one thing. Birds are pretty much doing one thing, but our squamate reptiles as a group are doing a lot of different things. And that's what we're of interest in today. But where does sex chromosomes come from, and why are we even talking about them? So to sort of set up for later parts of the talk, I need to tell you about sex determination as a process and how it's both extremely variable and also extremely conserved in vertebrates, because it doesn't make a lot of sense, but it's important to grasp. So this is a schematic of some of the upstream regulatory pieces of the sex determining cascade in mammals. And this is the most well studied. That's why we're using mammals as the model here. And you'll see that mammals have an XY system. This is conserved in all placental mammals. And this is the part where the sex determining gene, or SRY, the sex determining region of the Y, is located. This is the primary trigger for the sex determining cascade in mammals. This varies group by group. What that top trigger is varies significantly across the tree. However, this bottom piece, the thing that trigger initiates, is very conserved. And so we have this dichotomy of the top piece of the signaling cascade can switch over a bunch, but the bottom pieces remain relatively the same. And that will become more apparent or more important later in the talk. But I needed to tell you about that now. And what's going on with this variable part, because that's what we're going to be investigating more or less. So sex chromosomes, that primary trigger for sex determination, they evolve from an ordinary pair of autosomes. One of the two homologous pairs acquires a sex determining allele, and then throughout some evolutionary time period, recombination is suppressed around that allele, and that can happen through a variety of different mechanisms, none of which I'm going to be discussing today. But what's important to note here is that most vertebrate species occur in these sort of top two sections here, where the X and Z, or the Y and W, are indistinguishable under a light microscope. However, the most well-studied examples of sex chromosomes that we know of are these ones down here in mammals, birds, and even some of the lizards I'm going to be talking about today. Because most vertebrate species don't fall into this latter group, it's important to be studying these former sort of taxa. And both of the taxa I'm going to be talking about today, the Sphaerodactylus and crocodile lizards, are both in this former group. So we can't see these under a light microscope. We have to rely on the sequence information to study them more extensively. And so sort of as a feign of pitching my talk as sort of a hypothesis-driven talk, which I'm not really keen on, but here we go, are certain linkage groups recruited non-randomly to become sex-determining genes. So we have this pool of autosomes, and oftentimes when you look for tips across the tree, you see similar linkage groups evolving multiple times to be sex chromosomes. And the question is this random or not? Just as what the current state of the data might suggest, and I'm just going to read this quote for you from Consortium et al. A couple of years ago. The current data on the origin of sex chromosomes in amnio suggests that their evolution is indeed non-random. However, this non-random pattern is not very strong. So if we look at this as sort of a teeter-totter, sort of an analogy, it is teetering towards non-random, or it was a couple of years ago, but just barely so. And yeah, the p-value is not high for this, so and not a strong effect, even if it does come out in a model. And so today I'm going to tell you what I'm going to tell you, and then I'm going to tell you, and then I'm going to tell you what I told you. And what that means is I'm going to drop this weight called genomics on this right side of this teeter-totter, and I'm going to, with these two examples, show you that the more taxa we examine, the more likely it is that sex chromosomes are recruited randomly throughout evolutionary time periods. And so, like I mentioned earlier, we're zooming in on squamate reptiles, or this rainbow-esque pie here that I've circled. And if you zoom in on what that means, because this is the most speciose group of amniotes on Earth, there's a lot of species in this represented in that pie chart. And so here we have just a very large sort of graphically somewhat pleasing phylogeny of a tree of squamates, and labeled at each node and tip is the occurrence of either an XY sex determining gene or systems that have where the males are the heterogametic sex, ZW in orange, where the females are the heterogametic sex, and then temperature dependent in gray. And as you can see, some groups are more chaotic than others, and some of them are sort of all one color. This is outdated. Most of these have transitions that we didn't account for 10 years ago, but that's where sort of we're going to stand moving forward in the talk. And so all of that, what's important about that figure is it was generated without a ton of genomic data. Most of it was RADseq data, which I'll describe a little bit more detail later on, but we're missing a lot of information. Especially when you see the colors not changing in certain groups, that doesn't necessarily mean that they're the same system. That means that they're the same mating system type. Again, those are two very different things. And so to get at sort of where my work has taken me and where I've sort of had to overcome to get to what I'm presenting to you today, I need to tell you a little bit more about amniote diversity. So this pie chart represents the 4 major groups of amniotes. So we have 4. Normally I play a game. When I'm in person, I play a little game. I make people guess which pie chart is what. That doesn't work as well on Zoom, so I'm just going to sort of walk you through it. So again, there's 4 major groups. To orient you into this, this pie chart, I can tell you that the turtles and crocs are this small sliver here at the bottom, made up of 388 species total that we've currently counted. Next up is the mammals. You might be like, oh, there's a lot of mammals. There's actually not that ton of mammals, okay? So the best estimates I've seen so far are still less than 7,000 species. And these other 2 major groups, you might have guessed, are the squamates and the birds. So again, squamates are the most speciose group of amniotes on the planet, and they possess a ton of really interesting biology to study within them. Something like sex determination is one, reproductive modes, they have egg laying and live bearing, they've lost limbs dozens of times, they can regenerate their tails, they also lose the ability to regenerate their tails. It's a lot of cool things within this group that's worth studying. And yes, so let's compare to, oh, my little slide got cut off at the bottom. Okay, let's compare to what we might expect to see for genomic data in these groups, okay? So in sort of a neutral model or a null model, we might expect that the number of genomes being sequenced sort of mimics, even though the numbers wouldn't be the same, should sort of approximate the diversity numbers in each group. I'm going to tell you right now, no, doesn't happen. How bad is it? It's very bad, okay? So 15% of mammal species, so these numbers are a little off, I have to explain them. So the number is the number of genomes in that group. The percent is the number of species with the genome sequence out of the numbers on the right side of the graph. So 15% of the nearly 7,000 mammals have a genome sequence on, and this is just NCBI numbers in GenBank. And I ran these numbers earlier this week, squamates are the least represented vertebrate group as far as genomes go. So even behind amphibians, although the difference is negligible, they are underrepresented significantly in sort of the scientific literature. And this sets up why I'm going to be telling you about why we need genomes in squamates and some of the work that's being done in this group. So just running through this really quickly because it's not super important. Over the years, we had our very first genome published in 2011, and over the years, it was pretty stagnant up until about 2018, when we've seen a very sort of significant takeoff. And this has to do with a lot of things, but mostly the cost of sequencing has dropped significantly in the past few years. That has really allowed us to get more genomes for less money, because people studying squamates generally have less money than people studying primates, for instance, as far as research money goes. So getting back to sex determination and what today's questions are, today I'm going to zoom in on basically two nodes. One, and first, I'm going to be over here on the right side within the gecko lizards. At this node here and its descendants, this is the sphaerodactylid geckos that I'll be talking about. The second half of the talk, I'm going to be talking to you about anguimorphs, these all orange group over here, with possibly a temperature-dependent ancestor. So diving into today's data-driven part of the talk, the sprightly sphaerodactyl, and I'm going to explain why I keep using the word sprightly and why I will continue to use the word sprightly throughout the talk. So Sphaerodactylids is a family of geckos made up of 11 genera. We only know sex-determining information from 4 of them, one of which, Sphaerodactylus at the bottom, I'm going to tell you about today. Another one is Gonatodes, which that was part of my PhD work, a species that, again, lives in Colombia. And then Euleptes, which is occurs in the Old World, or Africa and Europe. And then Aristologer, which is another New World group. It's the only genus with the ZW that we know in this family. And so this enters the sprightly Sphaerodactylus. So these are my favorite species of lizard, group of lizards. This is the topic of my dissertation. So these are the sphero ball-toed geckos. There's over 108 species. They occur throughout the Caribbean. They generally occur in the leaf litter, but they do occur all the way to Colombia. There's a species that lives up in the bromeliads in the trees, and that's Heliconiae. They're also well known for this characteristic of being sexually dimorphic. The males generally have these really bright colored heads that are generally visually stunning. However, this has even been lost within the group. So all 4 of these species occur in Puerto Rico. 2 of them, all 4 of these animals are males. 2 are sexually dimorphic, 2 aren't. And I'm going to be talking about all these species today at some point in the talk. And so just giving you a little background as to the sphaerodactyls. A lot of the primary systematics done in the group was done by a former president's youngest grandson. And he had this really fun quote in one of his papers. And I'm not going to read the whole thing for you because it's quite long, but I will resonate with the very last sentence, and it is that I know of no genus that more amply repays finding, catching, and classifying than the sprightly little sphaerodactyl. And my PhD advisor and I thought that would be a lovely children's book, and so we commissioned an artist to basically mock up what the cover of that book might look like. And so this is just a general theme whenever I talk about Sphaerodactylus. I need to mention the sprightly sparrows. So in Puerto Rico, these are the salamanquitas. So there are no salamanders in the Caribbean native. And so they know what they're talking about when they say the little salamanders. But these are actually geckos. And there's a broad diversity throughout the Caribbean. The mainland and Cuban species have been severely understudied. But for the rest of today's talk, again, there's a lot of variation across the Puerto Rican bank. But for the rest of today's talk, I'm going to be parsing down this part of the phylogeny and showing you a much reduced representation. But know that there are other species to look at within this group, even though I'm not going to be showing them for the rest of the talk. So this is what that prune phylogeny looks like. So today we're going to have six species that we're going to be looking at. And to start, I need to tell you what we knew going into this project. And that is that 2 of these species have XY systems. And I'll show you here in a moment how I got that information. But we can see that both of them have genetic regions that are male specific out of these PCR gel validations on the right side. And so we got this information using a process called RADseq. And how we sort of make that work is we chop up the genome using a restriction enzyme. We can compare multiple males and multiple females to look for reads that occur in one sex and not the other. In an XY system, this is going to give you a lot of reads that occur in males and very few that occur in females. And what we could do is actually make PCR primers and validate that that region does occur in one sex and not the other. And the only region in the genome that occurs in one sex and not the other is the non-recombining part of the sex chromosome. And so we can use this as a validation effort and to confirm that we're seeing what we think we're seeing. And so we did this for all the species here, and we confirmed that all species that we're looking at that are shown here with a tick on the tree have XY systems. However, I can tell you we don't know anything more than this. We don't know if they're the same XY systems. So again, these would all be colored blue on the phylogeny, but we don't have any more information than that. And so the rest of the talk, I'm going to be telling you about what else is there to know. And so by only knowing that it's a blue tip, that can leave you with a lot of other questions. So again, this could all be an ancient conserved XY system within this group. So the same linkage group as the sex chromosome in all of them. That's a possibility. It could be a somewhat conserved. So the Puerto Rican species could have the same XY system, but it could be something different in our outgroup, right? That's a possibility. Or every species or every subgroup of species could be doing its own thing. Like all those are possible within all these tips are blue. And so to dive in deeper, we sequenced and assembled the reference genome for one of these species, Sphaerodactylus townsendii. And for the rest of the talk, or for the rest of this part of the talk, I'll be using them as our focal reference and moving from there. So what does it mean to have a high quality reference genome? It means that we can assign linkage groups or these chromosomal regions across evolutionary time periods. And so just showing you this ribbon plot linking chromosomes across taxa, you can see that there's a lot of rearrangements, at broad scales, but the groups themselves generally stay the same. And importantly, for the rest of this talk, both Sphaerodactylus townsendii and the second focal species I'm going to be talking about, Shinosaurus at the bottom, both of them have high quality reference genomes for us to use. And so, like I told you, we have this RADseq method to identify male or female specific parts of the genome. And for after we have a reference genome, we're enabled to do quite a few number of analyses. The major one I'm going to be talking about today is going to be this FST metric, and that tells us the genetic differentiation between the sexes at any specific region of the genome. And I will mention these other ones briefly, but FST is going to be the main one. And so, like I said, Sphaerodactylus townsendii is going to be our focal species because they're our genome animal. And so here at the top, we have across the whole genome, and the numbers on the Y value or on the Y axis aren't important. What's important is the relative number relative to everything else. Okay, so we have all of our chromosomes. We call them linkage groups because they haven't been physically mapped, but you could think of these as chromosomes if that helps you. And we see one region that is not like the others, and this is expected if it's the sex chromosome region. And to investigate this further, we zoomed in on linkage group 3 or chromosome 3, and just looked a little closer what's going on in there. So again, this is FST. This is the same metric, just for only this chromosome. We can look at our read depth. A lot of the times we see differences in read depth. We're seeing all of our reads map, suggesting this is a very young system. And if you have more questions about that later, happy to answer. And then in nucleotide diversity, we see that males have a lot more SNPs in this region than females. And all of that is very indicative of a sex chromosome system. And to really put the nail in the coffin of what I'm telling you is that we also can go in and say for sure that this region is sex-specific by doing PCR validations. And so now that I've convinced you that this is the sex-determining region in Sphaerodactylus townsendii, I'm going to sort of buzz through the other ones at a faster pace. The last little bit I want to tell you about this slide is we still have those sex-specific reads that were identified from the first part of the analysis that we didn't have a reference genome for. And we can simply take those and map them back to the reference and see where they land. And if we get a large cluster of those in a specific region, that's additional evidence that this is sex-specific. And that's exactly what we see here. So this rug on our plot here with the little orange dots, these are the mapped sex-specific reads from the first part of the talk here, and 87% of them map to this region. And that's really high. So now I'm just going to buzz through our other taxa really quick so we can get moving. So this is the same plot that we had on top in the last slide, just move down. I've also presented a phylogeny to keep you oriented as to what we're looking at on the left side. So if we look at the same thing in Sphaerodactylus nicholsi, its sister species in this context, we see 87 or 86% of those sex-specific reads are mapping to the exact same region, and we have elevated FST there as well. However, things start getting a little fun when we include another species out. So we see 2 whole chromosomes basically being incorporated into this region of high differences between the sexes. And one of them's on a different linkage group. Approximately 50% of our sex-specific reads map to one or the other linkage group here. So we're reasonably confident that both of those chromosomes are sex-linked in Anigoi. And I'm not going to talk much more about it other than the fact that subsequent work has confirmed that this is a fusion. So this is actually one linkage group in the species, but because we're relying on our reference genome, it's not fused. And so in this analysis, right, it comes out as 2 linkage groups. And then last is our outgroup. And this is a little bit more messy, but what we can see is a clear peak on linkage group 1, but not in the same area as Sphaerodactylus anigoi that I just showed you. So it's a little off-put in that. But 62% of our sex-specific reads map to that region there. And so we're pretty sure that we've identified the sex chromosome in all of these species. And so we can now take that information with the linkage and remap it to that phylogeny I showed you earlier on. And we can assign linkage groups to those XY ticks that we have. We can also then generate sort of going back in time information about how the sex chromosomes evolved. And skipping through, again, linkage group 3 is the sex chromosome in nicholsi and townsendii. We also looked in Cloudberry and confirmed that it's there as well. And so linkage group 3 was the sex chromosome probably in this ancestor. We see a fusion in Sphaerodactylus anigoi, and we still don't know what's going on in its sister species. And so the question then becomes, is linkage group 3 the ancestral sex chromosome in this group or not? And that's an open question that we are still actively working on. Given the reference genome and given high quality annotations or where the genes are within it, we're actually able to dig a little deeper and identify a putative sex determining gene within this genome. So like I said, the downstream sex determining cascade is relatively conserved. And so we do have this short list of genes that we look for when we're looking for sex determining genes. And one of them happens to be anti-Müllerian hormone receptor 2. And so that is located right in the sex determining region in our focal taxa. And so I'm going to mention this at the end of the talk again, but that's of interest to us. Again, another layer of things we can investigate once we have the genome. And so just running through conclusions of what I just told you, including a reference genome in this analysis allowed us to confirm or reject homology of these systems in some capacity. We identified between 2 and 4 turnovers within the system, just within the blue tips, right? So this is XY systems, but it's not the same system. And we're also able to implicate a putative primary sex determining gene, AMHR2. And so we're halfway through the talk of the data I'm going to show you. And what I haven't discussed is how this relates back to this hypothesis for today. And that is that both the linkage groups that we identified in these geckos as sex chromosomes have never been seen as sex chromosomes before in any amnio. So again, just a little digging a little bit deeper, we're getting just a much clearer picture. And this is going to be a theme that we're going to continue with for the next part of the talk here. So next is the crocodile lizard. And this one will go much quicker because I don't have to explain the methods to you again. So the anglomorphs. So this is a really sort of charismatic group of lizards. They're much larger than the last group. So this is the group that contains the Komodo dragon, the Gila monster, glass lizards. There's a lot of really interesting, cool species within this group, all of which I have basically the pleasure of working with now as a postdoc. So who is Shinosaurus? Why do we care? And I'm going to be saying Shinosaurus the whole time. It's a monotypic family. So there's only one species. So when I say Shinosaurus, I'm talking about the only one, right? I don't need to specify anymore. That's who we're talking about. So I will be saying Shinosaurus for us to talk. This is the crocodile lizard. It's endemic to southern China and Vietnam. It's very endangered. It's semi-aquatic, relies on pure water streams to survive and reproduce, and those are disappearing for some reason very rapidly. Importantly for today's story, they're sexually monomorphic and incredibly difficult to tell apart. There's maybe a handful of people who can reasonably confidently sex these animals on the planet. However, captive breeding in zoos at a higher rate is necessary for this species' survival, basically, because its habitat is being destroyed faster than we can do anything about it. And so in order to more effectively captive breed, wouldn't it be nice to know what the sex chromosome system is so we can sex them genetically? And that is actually the justification for why we even started this project to begin with. So we know that anglomorphs possess an ancestral ZW system located on the syntenic region of chicken chromosome 28. And this occurs, again, all in these blue taxa across the phylogeny. There's a lot of unknowns here, but what we do know is that Shinosaurus probably doesn't have that. And so what does that look like? This is just a reminder that this is homomorphic and we can't see it under the light microscope. But that sex chromosome system that occurs in the Gila monster and the Komodo dragon, we can map it to the Shinosaurus genome. Shinosaurus has the best genome of any anglomorph, so that really helps us specifically for this question. But we know the sex determining scaffolds in those more draft assemblies, and we map them to the genome, and they all map to this distal end of linkage group 7 or chromosome 7 in Shinosaurus. And so we know if the sex chromosome that we identified doesn't occur on linkage group 7, then we've identified a turnover to a new linkage group. And so just a brief overview of what we're doing. Again, we're going to do whole genome scanning using FST again, like I did in the last part of the talk. We have more individuals here for a variety of reasons, and so we're going to identify SNPs that occur in one sex and not the other. And then, again, we're going to dive into identifying some candidate sex-determining gene using this genome. And so the data we have available to us, we have an old reference genome for a male. We have a female reference genome. This is the new one. It's really good. And then we have whole genome resequencing data for multiple males and multiple females we'll be using for identifying. And cutting straight to the chase, this is a ZW system. There's a large uptick of female-specific SNPs located on linkage group 3. Mind you, the ancestral chromosome is over here in linkage group 7, so that's not what we're seeing. This is a transition to a new linkage group. And this coincides with elevated genetic differentiation between males and females. So this appears to be the region that's determining sex in the crocodile lizard, Shinosaurus. And the more interesting part of this part of the talk is actually what's in that part of the genome. And so we identified the genes that are in there. It's less than a megabase, so it's less than 1 million base pairs long. There's only 10 genes in there, which makes it a really short list to identify which gene might be determining sex. And like I told you earlier, the downstream part of the sex determining cascade is very conserved in vertebrates. And one of those genes that's very conserved and interesting happens to be this one, FoxL2. So I need to describe to you a little bit about what FoxL2 does and why it's interesting. And so that I have to go back to the first part of the talk. So this is the picture I showed you at the beginning. So FoxL2 and DMRT1 are famous in the sex determining literature for a variety of reasons. You'll notice at the bottom of this figure, DMRT1 is here on the left, on the blue or the quote unquote male side. FoxL2 is on the other side, on this pink side. So FoxL2 and DMRT1 are sex determining genes in all vertebrates. FoxL2 and DMRT1 antagonize each other. High expression of FoxL2 represses expression of DMRT1 and vice versa. And DMRT1 has been extensively studied. So understanding what DMRT1 is doing is going to provide us some insight as to what FoxL2 might be involved in. And importantly, DMRT1 is a sex determining gene in a ton of species. In all birds, it is a sex determining gene. In Medaka fish, it is a sex determining gene. And in Xenopus frogs, it is a sex determining gene. So this gene it's a recurring character in this story of what are vertebrates doing in sex determination. However, FoxL2 has never been identified as a sex determining gene, and this has always been sort of an oddball thing to see, right? Because it's just as important as DMRT1, but we haven't seen it yet. And why that is, who knows? But we're going to start maybe today with figuring this out. And so, like I mentioned, DMRT1 is very well characterized. And I'm going to explain to you in all species what the current model is for how DMRT1 becomes the primary sex determining gene. So on A here in mammals, that's just sort of what it does generally, because it's not the primary sex determining gene in mammals, but it's involved. And so that's just sort of our baseline. In Medaka fish over here in this box, DMY is a DMRT1 duplicate, and DMY is expressed earlier than autosomal DMRT1. So they have both copies, and it's what do you say? It helps itself amplify itself. So it's a feedback loop, right? Okay, sorry. So it feedbacks on itself positively. And so it gets expressed a little earlier than autosomal DMRT1 and activates autosomal DMRT1, which initiates basically testes differentiation. In birds, the W copy or the W allele is missing DMRT1, where the Z has it. So in females, you only have one copy of DMRT1. That reduced expression of DMRT1 initiates ovary development. In males, when you have two Zs, you have two copies, you get double the expression of DMRT1, it initiates testes development, right? So we're getting this theme of high DMRT1 equals maleness or testes development. And lastly, in Xenopus frogs, the W copy of DMRT1, which is a truncated version of the gene, interferes with the autosomal copy. It gets expressed earlier, just like DMY does in Medaka. However, it's truncated and it can't do its job properly. So it actually results in lower functional DMRT1 expression, leading to ovary development. So again, there's multiple ways in which DMRT1 can be involved, but DMRT1 expression needs to be lower in order for femaleness or ovary development to initiate. And like I said, FoxL2 on the right and DMRT1 antagonize each other. And so FoxL2 can be one of these mechanisms that lowers DMRT1 expression, although it's never really been seen before as a primary trigger. And so if FoxL2, our gene of interest, is determining sex, we need to develop some sort of a model that implicates that FoxL2 is likely expressed higher in females, leading to the reduction of DMRT1 expression in females, initiating ovary development. Okay, and so that's sort of how I've expressed it here. Again, these are antagonizing each other. And so do we have enough information to implicate FoxL2 in a sort of an informed way, knowing what we know about DMRT1? And the answer is, what is that block of white doing in the middle of my table? And it is a FoxL2 duplication that occurs only on the W chromosome. And so this gives us a model where there are extra copies of FoxL2 in the genome, in females specifically, on the W, that could be this amplified FoxL2 expression, which would repress DMRT1 and lead to ovary development. So, and this duplication is specific to Shinosaurus recently, which would again implicate that this is the sex-determining gene, or at least is heavily involved some way, somehow, in sex determination in Shinosaurus. And so we can add that to our very short list of known sex-determining genes, or implicated sex-determining genes in all squamates. There's only 4 of them, 3 of which are in anglomorphs or sphaerodactylus geckos that I've talked about today. And then there's the bearded dragon, which is doing something different. But again, just by having this reference genome, we're getting a much more refined look at what we're seeing in what we're missing. And so to wrap up this last part of the talk here, by incorporating a reference genome into sort of already existing data, because we didn't have to generate additional data for this project specifically, we identified the first confirmation of a ZW to a ZW turnover in anglomorphs. And this will hopefully allow folks to sex animals genetically during their captive breeding efforts, and hopefully will have some positive impact on their ability to survive the anthropogenic era. This is also the first implication of FoxL2 as a primary sex-determining gene in a vertebrate. And last but not least, this chromosome that we discovered this gene on that's never been seen is also a brand new linkage group that's never been seen before as a sex-determining linkage group in a vertebrate. And so what we can do is we can finally drop the genomics weight on this right side and send this non-randomness thing sort of off into oblivion for the rest of the talk, and suggest that indeed, the more we look, the more likely we are to find that these things are random. Sex determination, like other things, is not a it's not a directed process. There's no end goal. It is doing what it's doing. It is being allowing organisms to survive and reproduce. Right.
We have 8 minutes left, Brendan.
Sounds good. Thank you, Sarah.
All right.
So I told you what I was going to tell you. I told you, and now I'm going to reiterate what I told you, running back through my outline. So today, to start, I told you about sex determination. We started at base pairs of DNA, how chromosomes evolve, where sex chromosomes come from, and how they determine sex. And then we use this information to investigate Sphaerodactylus geckos from Puerto Rico and the dynamic sex chromosomes in that system. And then we took that information and adapted it to the crocodile lizard from China and Vietnam. And I hopefully got across that the incorporation of high quality reference genomes was vital to both of these investigations, and allowed us to really dive in to see some more interesting things that we didn't know existed within a system that we thought we knew, right? So future directions for this work. So Arizona is home to a lot of cool lizard diversity, 2 of which on the right here, the Gila monster and the alligator lizards occur. Both of these are anglomorphs, allowing us to extend our study of Shinosaurus lizard sex determination across the anglomorph tree, just within Arizona. And so we're going to be sequencing their genomes here very shortly. And Arizona also has 3 species of night lizards, another really interesting sex determining species that we picked up some preliminary data for during my PhD. And then we have one species of endemic gecko that doesn't have a genome and has interesting sex chromosome things going on, and we'll be sequencing them as well. In fact, later today, I'm headed to Tucson, and I will be scouting out some places to potentially collect these guys just in a few hours. And so I mentioned earlier that squamates are the least represented group of vertebrates as far as genomics goes, and that's due to a lot of reasons, but the reasons are not as important as changing things for the better moving forward. And in an effort to do this, we've recently started the Open Consortium of Squamate Genomics. This is an organization that theoretically will allow everyone everywhere to be not only doing genomics in squamates, generating the genome sequences, but to be communicating with each other so we're not wasting money, time, have a resource for asking questions to people who have done it before. There's no reason for everybody always to be reinventing the wheel, especially in a taxonomic system where there's not a lot of people doing it already because we don't have a lot of sequences. And so who are you going to ask if you have a question? And there's not that many. So through sort of training and collaboration efforts, we're trying to sort of rally the troops to increase squamate genomes as a resource for sort of investigating not only squamates themselves, but genomics across all vertebrates. And so that is exciting and a new thing I've been working on. And with that, I will take any questions, and thank you so much for your time and attention, and hope you enjoyed my ramblings about geckos and crocodile lizards.
Thank you, Brendan. It was super interesting, and it always makes me think a lot the amount of diversity we have, not just morphologically, but just how reproduction and sex comes about. I have one question, and then we have another question in the chat.
Yeah.
I'm always very interested about how do you teach to students, young or older people, about sex determination? Like, we're all fixed on like Y and X, and maybe some people know about chicken system, but then this thing is just, it's just crazy to me. When you start teaching this to people, how do you approach that? How do you do this?
I think number one, and sort of, especially depending on who your audience is, and if we're talking about like an undergrad who may or may not ever take a science class again, and what you're trying to get across to them or something like this, I would say rejecting the idea, getting across that you can reject the idea that there is a normal.
Okay.
There is no normal, how evolutionary speaking, there's no normal sex determining way, right? So across all, there's species that determine sex using mitochondria. There's species that determine sex using 56 different sex types. There's so many things going on out there, and we've been conditioned socially to think that it's a binary, and I would suggest that we reject that. And when you do talk about sex in any specific way, qualify what you mean by that, because it can mean a lot of things. Biological sex is not a gamete. It's not a reproductive part. It's a lot of things, right? And by just and I hope I did this reasonably well during my talk, is when I talk about developing ovaries, that's what I mean. I don't care about other parts of their biology. I'm concerned with that. Who's carrying the egg in a squamate? Who's carrying the sperm in a squamate? That's all I'm concerned with. But there are so many other aspects there,
right.
and sometimes we conflate them. And I would say really getting across the point that these are not correlated or associated at all. Yeah.
Right. Thank you, Brendan. Okay, we have one question in the chat from Juliana. Maybe you want to see it yourself, and I'm going to read it regardless. So,
tenemos dos preguntas en el chat.
sí, tranqui, voy con la primera.
Okay. Listo.
Okay. The first question is, do you know if other genes linked to the sex determinant locus are involved in the same pathway or similar pathways to that of FoxL2? Otherwise, do you have any explanation as to why selection might have favored their clustering?
No, none of those other genes seem to be involved in sex determination more broadly. Most of them don't have names, so that sort of precludes our ability to even identify what they're doing. But to the second part of the question, why FoxL2 appears to be clustered there, that would be sort of a very understandable thing to happen if the gene is just duplicated right next to itself. It would be, again, there's more process involved if you're going to take a gene and move it to a different chromosome, but it's actually a fairly simple mechanism to just duplicate that region at a very proximal scale. And so that's what we think has happened. Although you may have noticed that it was a female reference, and so the Z and the W are both sort of collapsed into each other on that reference genome. They didn't know that it was a ZW system when they sequenced it, so they wouldn't have known during the assembly process what to look for.
Okay. All right. The second question is from Camilo, and he left, so I'm going to ask it regardless. But Camilo's question is, do you think that the two XY systems you found are related? I mean, do you think one of them is a new sex chromosome that arose from the other?
That's certainly possible. The out group is different, right? Because it's just in a different part of the linkage group, and so we can just sort of
cross that out,
eject that. Yeah. But within our focal group with an ego eye, which is, again, this is the picture for them, with that has the fusion across the two chromosomes, and then our two other species that had just that spike. The ego eye system does encompass where that spike occurs.
Okay.
And so it's possible that that's the spike system is the ancestral state, right?
Okay.
That was the sex chromosome, and it's just been expanded in an ego eye. However, we've looked a little deeper in that region, and we don't see the same signatures in that spot in an ego eye, which we would expect to still be present regardless of if it expanded out.
Okay.
And so either when those species diverged from each other, that system was very, very young, and it just hadn't done a ton of degeneration, is what we call the process of things getting different between the X and the Y at that point. And so it wasn't present in their common ancestor. That's possible.
Okay.
But we don't know for sure. So
got it. All right. Do we have any more questions from the audience that are not in the chat? No one else. Okay. I'm going to take the liberty to go one more minute. Okay. Why do you think then there is just more money to study mammals? Just because, I mean, people are not interested in lizards and reptiles and crocs in general. I mean, why so few genomes? I mean, are they hard to, I mean, is it just amount of money, or is it just hard to sequence genomes of squamates versus genomes of mammals? No, it's the same.
no, it's actually much easier to sequence squamates.
Oh, wow. Okay. So
Yeah.
this is just, we're more interested in mammals as mammalians than other groups.
Yeah, our hypothesis, and it's really hard to test this, you know, this is it's a, you know, more of a social issue than it is a biological one. But what we did is I looked at a large subset in a recent review of where the funding came from for these for a bunch of mammal genomes and squamate genomes.
Okay.
And the funding for mammals comes from the National Institutes of Health.
Right. And
those grants are enormous.
Large. Yeah. Okay.
All the squamate genomes, maybe sub-1, was funded either by personal funds.
Okay.
Or the NSF.
Right. Okay. So it's
and
just
those grants are much smaller. And again, personal funds, laboratory startup. I actually paid in person for like myself for some
I
of the
know.
sequencing in my dissertation that
I know.
happened.
I
I know you
remember
did.
that. Yeah.
But again, so the money just wasn't there. And
Okay.
that being said, they're getting there. The NIH is understanding now that all species are more important than they were giving them credit for.
Okay.
So
Got
we
it.
think it's going to change, but we're not there yet. And the numbers haven't caught up
Right.
with things. So
Sure. All right. Brendan, thank you so much. I think it's super interesting to think about sex determination and how the diversity of not only sex determination systems, but just diversity as a whole is just so many layers within, and thinking about the genes themselves and how, where are they located in chromosomes. I think this is fascinating. And thank you so much for answering our call to give us a seminar. I want to thank everyone else for attending. I'm sorry that we extended a little bit the presentation. Thank you again. Gracias a todas y todos por venir. Have a great weekend. Thank you, Brendan. Good luck finding those geckos. It's going to be a fun Friday.
thank you
All
so
right.
much for having
Thank
me.
you
I
so
really
much.
appreciate it.
Yep, no problem.
Okay,
Thank you, Brendan.
Bye, everyone.
Ciao.
bye. Gracias. Buena tarde para todos.